Good evening. Well, I tell you what, we've been on a journey these last 5 nights, haven't we? I think we all can confidently say we have seen the complexity of the human body and some amazing things that a divine creator had his hand on. And it is just simply awesome to see all those bits and parts. I was truly impressed last night with seeing the stages of birth. I mean of the growth of the baby, and oh my goodness, I tell you what, that is simply amazing. God is so amazing. We are wondrously created, right? Jim, thank you so very much for coming out here and sharing this with us. We truly have been blessed, and we're definitely going to get you back out again. So, but before we start off here on our last night, let's bow our heads in prayer. Father in heaven, Lord, we just truly thank you for this time again that we can come together and we can see the wondrous works of your hands, and to see all the complexity and all the details, that you're a God of order and structure. And so, Father in heaven, as we dive in tonight, we just want to say thank you. Thank you for the fellowship that we've had with our friends. Thank you for the fellowship and the learning that we've had with Jim and his wife, Neva. And thank you, Father, for this opportunity that we can come together in this place. And so, Lord, we just ask that you'll bless us tonight as we continue on with this study. In Jesus' name, amen.
It's been special for Neva and me to be with you. Normally when we travel like this, she has to work the whole day preparing the food for that evening. And so she really loves it when we do this thing instead of the other thing. And on those other occasions, I have all day to play around. But for some reason, it has not been the case this week. The problem is I keep revamping this program, and so I've spent a lot of time doing that. Which is not maybe the best news. Maybe it is, I don't know. But this program is available for purchase. A couple of people have asked me about that. What's wrong with that side of the church? Is there something over there?
That's the left side.
Cold over there. You're going to get a complex tonight. Anyway, if you're interested, you can get on our website and purchase the 5 DVDs that constitute this program. The interviews are the same, but I never do 2 presentations the same anyway, so I guess that's just the way it is. I didn't bring many of these. I don't know that I even brought over a dozen, but in case some of you are interested in, either you know somebody that needs help getting well, or you might have an interest. This is a little flyer out there about our work in Spokane, if you're interested. A question came in regarding the chromosomes, and so here's a little clarification maybe. This one, let's say, came from the father, this half. So in the sperm there's 23 halves, and in the ovum, the oocyte or the egg, all of those are proper terms, is the other half. And of course this is coiled, wound, folded, and coiled again. You all remember that, don't you? And there are 23 chromosomes. Some of them are shorter, some of them are longer. And so I've always talked to you about the ladder as though they were all hooked together, but they're not. They're in these pieces. But if you in your mind hooked all these pieces together, this would be the total genome. It's interesting that one of the ways, when I said hooked together, I hope that made sense. Like just one after the other hooked together would be the whole genome from the father, his half, and the other from the mother. It's interesting that one of the ways that corrections are made when there is copy error, remember that story, is that the proteins actually compare. And they look at this one and fix that one accordingly. That doesn't mean that the two are now exactly alike. Nobody understands how this works, but somehow they keep intact the individual characteristics of the parent and the two parents. But that's one of the ways that the copy error is repaired. It's a very elegant thing, and there's a lot we don't know about it. Now, it's interesting that we draw this picture, but the chromosomes only look like this for a few seconds while the mitosis, that's the word for dividing the cell, takes place. And within moments after that, they change their configuration, and you look at the, you can actually look at pictures of this online. There's no resemblance to the chromosomes that we like to draw, and actually we label where the genes are. We have a system of labeling. This would be the P, and it would have a number. P means this particular piece up here, and it would have a number, and so forth. So you can, just by looking at this, know where to look on the chromosome for this particular gene that they're talking about. Anyway, for what it's worth, that's a little bit more of a primer on chromosomes. I want to take a few minutes this evening to start out with with not an interview that I did, but an interview with David Berlinski. You'll have to tell me what you think of this. I've never used him before, although I just am enamored with his intelligence and his capacity to articulate things. I talked to you about that a little bit. The first 10 seconds or so, it's just giving his credentials, which are pretty interesting. And then he is speaking to somebody and answering questions that will be put on the screen. Let me make this as full screen as I can and see if we can hear this. Raised in New York City, educated at Columbia, received a PhD in philosophy from Princeton, became a fellow of the Faculty of Mathematics and a postdoc in molecular biology. He's been a fellow. This means like another doctorate, really, in the International Institute for Applied Systems Analysis. And on it goes. Critically acclaimed documentary, Icons of Evolution. He's in that. How many ever saw this thing called, by Ben Stein, what is it? Not too many. No, it was like, not excommunicated, but left out. What's the word? Anyway, yeah, it was a DVD out there. It was a DVD that looked at how the scientific community doesn't want to answer these tough questions. Anyway, he was one of the people interviewed in that film, and so here he is. Incomparable. They use a bunch of these I words about him, and they're really kind of all true. It was earlier. Remember when I was playing the music?
Oh, okay. Well, in 1973, I was a postdoc in molecular biology at Columbia University. And it was a time really of a lot of intense discussion. I had a lot of friends who were postdocs. Subjects came up naturally. And I just happened to come across a copy of the Wistar Symposium that was held in 1966 in Philadelphia. It's a collection of essays about Darwinian theory. And I read Murray Eden's article, a critical article about Darwinian theory, and Marcel Schutzenberger's critical argument about Darwinian theory. And I started talking about it with the other postdoctoral fellows, people who were working in the laboratory at the time. And I discovered, somewhat to my own surprise, that the arguments, which seemed so very credible, very important, went virtually unanswered among the biologists that I knew. who tended to dismiss the arguments in a way that suggested that they hadn't really understood them, and if they had understood them, were not prepared to respond to them. And that was the beginning of my skepticism about Darwinian theory. When I spent a year in Paris working with Schutzenberger, of course, both of us enriched each other's opinions. Schutzenberger had been a long-standing critic within the French biological establishment of Darwinian theory. And what he had to say reinforced what I had to say, what I had to say reinforced what he had to say. Later I talked with Murray Eden. There were a group of us who were similarly skeptical. I must say in the 70s, in the late 60s and in the 70s, there was a much more intensive degree of opposition to Darwinian theory, a much greater willingness to examine Darwinian orthodoxies. The great counter-reformation took place in the 1980s and the 1990s. So when I started work, or when I started thinking about these issues, Schutzenberger and I wanted to write a book together on this. There was a very relatively liberal attitude among mathematicians, people who were interested, physicists, people who were interested in Darwinian theory, a much greater willingness to wonder whether any of this could possibly be true. So that was roughly my own background, my own approach to it. Well, the claim that all skeptics about Darwinian orthodoxy or Christian fundamentalists stands refuted by me. It's obviously not true. I'm neither Christian nor a fundamentalist. But lots and lots of people are skeptical in the scientific community. I know dozens of mathematicians who scratch their head and say, you guys think this is the way life originated. It's absolutely a preposterous theory. And many, many very significant figures. John von Neumann, one of the great mathematicians of the 20th century, just laughed at Darwinian theory. He hooted at it. So it's perfectly absurd. This is a point in a polemical dispute. It's not a reasonable standard of criticism. Opposition to Darwinian theory is, I wouldn't say widespread, but there's a consistent group of people among mathematicians, among physicists, among some very good speculative biologists who simply don't accept it, don't even regard it as a scientific theory in any reasonable sense. It's tough to say because we're not dealing with a theory in any sense in which, say, a physicist would recognize the theory. We're dealing with a collection of anecdotes, a certain point of view, a series of hunches. I would say that the most outstanding, the salient points are, first of all, the fossil record, which is simply mystifying. We can't make much sense of the fossil record. It does not sustain any kind of Darwinian prediction that can be intelligently derived from Darwinian theory, and it doesn't seem to sustain anything else as far as I can see. It's a perfectly mystifying record. That's one obvious point. I'm not talking just about the Cambrian explosion. I'm talking about everything that doesn't make a whole lot of sense in the fossil record. Second point, we have never been able in any way theoretically to examine the central Darwinian claim that natural selection and random variation can account for a great deal of complexity. If you look at the history of physics, for example, what did Newton do in the 17th century? He said, well, the planets are being attracted to the sun by a force. It's not any kind of force, it's a inverse square force. And then he went and showed that if you make that assumption, the result will be an orbit that conforms exactly to the observed orbit, say, of the Earth or of Mars. It will be a conic section. And then he proved the converse, that if it's a conic section, the planets must be attracted to a central source by an inverse square law. There is nothing like that in biology, in Darwinian theory. A kind of a canonical demonstration that this mechanism, random variation, natural selection, is adequate to the generation of this level of complexity. From the point of view of the serious sciences, without that kind of a demonstration, one is completely adrift. You have no idea whether the mechanism is adequate for its intended purposes. This is the second point. Third evidential piece of the puzzle. Look, you turn to the serious sciences, you turn to general relativity or quantum mechanics. I can program a computer with the equations of general relativity or with the equations of quantum mechanics, and I can say, all right, what are the consequences? I can actually see the consequences emerge in a simulation. We can't do any of this in biology, and that should prompt any reasonable person to ask, why not? If this is such a simple mechanism, which which can easily be programmed on a computer. How come we can't set up a computer and create something of biological-like complexity? How come we cannot see the unfolding of an evolutionary process the way we can see the unfolding of an evolutionary process in physics? This is a very serious question. I've looked at all the genetic algorithms. I'm trying to write a genetic algorithm myself. And the sheer fact is, without a tremendous amount of very special manipulation and ad hoc constraints, the computer is not going to generate anything realistic if it uses Darwinian mechanisms, and it will generate something realistic only if it doesn't use Darwinian mechanisms. This is an important point. 50 years after the computer revolution began, we have a splendid tool for assessing the intelligibility and viability of Darwinian theory. And everything that we know, I think this is the uniform experience of anyone working in genetic algorithms, indicates these mechanisms will not work. They will not work for their intended purposes. And finally, there's the utter absence of laboratory evidence. I mean, random variation, natural selection, we should be able to start manipulating organisms. When we look at dogs, no matter how far back we go, it's dogs. When we look at bacteria, no matter what we do, they stay bugs. They don't change in their fundamental nature. There seems to be some sort of an inherent species limitation, and we have no good explanation for this in terms of a Darwinian theory. We should have far more flexibility, far more plasticity under laboratory conditions than we actually do if Darwinian theory or anything like that were correct. What we see in nature, what we see in the laboratory, is very highly bounded variation, cyclic variation, as for example, finch beaks in the Galapagos Island. That's about all we see. Small variations. Why is that if Darwinian theory is correct? These are evidentiary points that I think need to be stressed, need to be examined openly, honestly. And they never are, of course. Never are.
Now, you might want to listen to that about 15 times before you could really get it all, right? But could you sort of tell?
Absolutely.
There was serious thinking going on there, wasn't there? And just to catch you up a little bit, in case you haven't read this or known this, when Darwin was on the Beagle for that, what was it, several-year tour, and got to the Galapagos down there off the coast of South America, among the things he studied, and I said the other night, I think I said this, Darwin was a very brilliant, smart fellow, and he amazes me at how detailed he was in taking data and in the observations that he made and recorded and so forth. But he noticed that the finches on the island, and this was over some period of time, their beaks changed. They got longer and more pointed, and this was one of the things he felt was showing that there was evolution taking place. What he didn't realize right away was that Berlinski said this, it looks like it's seasonal, because when it gets dry, then their beaks get short and stubby again, because where they have to peck to get their food, I can't remember what it is, it's not a pine cone, but something like that, some kind of a nut or something, they get there, it's tougher to get it out, and they got to have a tougher beak, and so their beaks change. And that's what he was saying. All we see there is cyclic variation that apparently may be built right into the genome. My own personal belief is that the original genome for the original dog had all the dogs in there that we know today. Those dogs aren't the result of natural selection. The genome has that capacity. I've got to be careful about taking comments, but
An observation from what you just said. It seems like it's not so much science, but more like a religion.
well, it isn't science. That's the issue, and some people speak of it as a religion. You'd have to understand what the definition of religion would be. They're certainly not worshiping a supernatural God, which normally we think of as part of, but religion could be, defined on a much broader basis if you wanted. All right, I think I saved the best till last, with one possible exception. We would, if we knew as much about the brain as we know about the eye, that would be really fun. But that was kind of Ben Carson's territory. So I want to look with you this evening at the eye. And even though this comes later, I wanted to get this into your mind. You've all heard about MS. I think you probably all know that there's something called a myelin sheath on the motor nerves. And as that sheath is degraded by the immune system, that's what's happening. The immune system is attacking. The conduction of the signals doesn't take place properly. And the way this works, in case you haven't seen this, is that what I showed you, the dormitory-like propagation of in and out with the little electric charges, would proceed relatively slowly. When I say slowly, it would take a couple of milliseconds to get from one end to the other. But when the myelin sheath it grows, is placed, if you will, with these nodes between these sections, the propagation of this signal doesn't have to go tick, 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 tick. It jumps, the effect jumps from here to here almost instantly, almost at the speed of light. And so so the propagation is extremely fast down the neuron to the muscle that it's being operated. Now, in the eye, as we will see, there's a huge amount of nervous tissue, but none of it is myelinated. After it leaves the eye, the optic nerve is myelated from them on. So we'll kind of get to that as we go along here. Well, here's a diagram of the eye, and you all know quite a bit about the eye, I'm sure. This is the cornea, and the cornea causes light. Excuse me for this, using this term. I guess I left the black pen somewhere else. The cornea causes light to be refracted, which just means bent. So I should have just said it makes the light bend. And that's, of course, in order to focus the light back on the retina. And so it's something like this. I'm going to draw a simpler diagram to illustrate something else here in a minute. So here's this lens, which is partly the cornea, and then the thing in the eye that we call the lens. It's not a very good representation there. Light's coming along like this, and in the lens it gets bent. Actually, it gets bent twice, going in and leaving, and so it gets focused out here somewhere. We say focused. And that's why we get a picture in focus on the, if I can see if I can hit the same dot, to focus it, in this case, on this curve that is the retina. And all of you know, I think, that there is this place where the optic nerve forms, and we'll see this in a minute, from all of the nerves around the retina. They come together here. There's 1 million nerve fibers that come together and exit through the back of the eye and then out through the back of the bony place there. And you don't have any vision right there. You all know that, right? If you want to test it just for fun, if you hold your thumb about this far in front of your nose and cover one eye, and if you look straight ahead and don't quit looking straight ahead, and as you move your thumb away, the tip will disappear right about there. Now, because your whole thumb is visible, it might fool you for a minute, but the tip will disappear. You all have tried that before, haven't you? Everybody's got their thumb up now. The so-called blind spot. Of course, when both eyes are there, you can't see that. In any case, the cornea bends the light some, and then the lens bends it some more. The cornea actually bends it more than the lens, but it needs both in order to focus the light back here on the retina. And the retina is the main thing that we're going to look at this evening. It is just so amazing how this whole thing works. There's another picture of the eye, and the artist has taken a little bit, this brown layer is the retina, and he's taken that and he's tried to draw here the retina. The retina has 10 layers of cells, and it is about the same thickness as saran wrap. 10 layers of cells. And we'll look at that just a little bit here as we go along. This is several layers here. All of them are not shown. Interestingly enough, Darwin, in one of his books, I can't remember which one it is now, says that evolution did a pretty poor job of making the eye because the retina is kind of backwards. And the reason he says that is that the light-sensitive elements, the rods and the cones, are down here almost at the bottom, if you want to call this the top and this the bottom. And there's a lot of nerve cells here, and their connections all come up here and run sideways over to the optic nerve. You following what I'm describing? And so not only are all of these nerve axons in the way, but so are all these cells. And it's very interesting that the Creator used this system. Here's another drawing. I'm sorry, it would make more sense if the light was coming from the top. But in this drawing, the light is coming from the bottom. And this is a layer of cells that's kind of the bottom of the retina pigmentosa, or they call it the pigmented cells. You can see little cells there, each little block. And this is a cone, and this is a rod, and the light's coming from down here, and it has to pass through, and it doesn't even show. Well, it does right there. The artist is trying to show some lines there that are all of the nerve fibers traveling off, I guess, in this direction over toward the optic nerve. And this is an amazing thing here, and I'll show you more in a minute. But we have these ganglion cells, which are actually nerve cells, and we have a whole set of cells called bipolar cells, which are on and off switches. As the light is coming to your retina, this image out there, the eye is rapidly deciding which light ray to allow to make a cone or a rod sense the light and make a nerve signal. There's a bunch of on and off switches going on in there. There are 126 million rods and cones in the retina of your eye. And from those, and the ganglion cells, and the horizontal cells, all these nerve fibers, a million nerve fibers come out from all of that to go down the optic nerve. And it's very much unlike a camera. Most of you probably know this, but when you buy a nice camera, nice or not, it's the same thing. Today, instead of film, there is a semiconductor surface that's sensitive to light. And they make it today, the little tiny pieces that are sensitive to light are very small. We call them a pixel. You all know that, don't you? And every pixel that receives light on that little semiconductor or chip is a pixel on your screen. One-to-one relationship. And the way it gets color is there's actually 3 pixels for every pixel. And one's red, one's green. One receives red, one receives green, one receives blue. And on your television screen, if you got a magnifying glass and went up real close to it, you can actually see the red and the green and the blue dots. They're very close together to make sort of one dot. And today we have resolutions on television screens that are, 1,700 dots per whatever inch or something. So it's very high resolution. But that's not the way the eye works. In fact, we don't know how the eye works. As we'll see, the signals coming from the retina eventually end up being an image that we see and take for granted. Most every day I go the whole day and I never think about the fact that my eyes are doing this for me. We just open them and use them. The retina's inverted structure, that means this was this was Dawkins' comment. It seems like it ought to have the light receiving things out at the front. So that's what he means by inverted structure. You all with me? Now, Joe told me every night he listens to me, and he says, every time I say, are you with me? He turns around and looks at you all, and he says, they all look, their faces are all blank. I assured him that you really were getting it, so he just wasn't sure by the look on your face that you were. All right. The retina's inverted structure might seem ill-suited to its function. The rods and the cones are the photosensitive cells that transduce light energy. That means change it into an electrical impulse. And they actually point away, look at this, they point away from the light. The light sensitive portion of this are, and we'll talk about this in a minute, are little, I'll show you a picture actually. Can you kind of see some horizontal lines there? There's hundreds and thousands of discs, they call them, in each of the rods and the cones. And that's where the light, when it hits, chemistry takes place and makes some electricity that ends up becoming a nerve, a signal on a nerve cell. In fact, the cones and the rods are nerve cells, the light. Boy, some of you are scaring me. Several of you are thinking with your eyes closed. Boy, I wish you wouldn't do that. It gives me the impression that it's not important. And anyway, so back to what he said. They transduce light energy into electrical impulses, and they point away from the incoming light. And they're located at the back of the retina, so that light entering the eye has to pass through several layers of cells before it reaches them. The retina also contains nerve fibers that are positioned perpendicular. Remember, I put in the word crossways. To the path of light entering the eye, and many of the structures in the upper layers have a diameter as big as the wavelength of light, which means that they could interfere with the light. If they were a lot tinier than the wavelength, it wouldn't matter. That's the idea there. Because of this inversion, one would think that incident light entering the eye should be subjected to a significant amount of reflection and scattering, yet the retina performs its function perfectly. Pretty interesting, wouldn't you say? Now let me give you another primer here that will help us as we try to put our heads around this idea. You probably haven't thought of this unless you took physics one time in your life, but if there's a lake surface here and you're swimming along here with your scuba gear or whatever, and you look up, you can see the sky, of course. But if you keep looking closer and closer to the shore, let's say, there comes a point where you can no longer see out. It's like the light ray, if it was leaving your eye, which it isn't, would hit the water and it couldn't get out, and it would just travel along the surface. And if you increase the angle some more, it actually completely reflects back inside. Did you know that? How many of you knew that? Two hands, called total internal reflection. And fishermen all know this. Well, at least they kind of know it. They know that if they stay back a little way from the water, and if they stay low, the fish can't see them. Got it? And this is, I won't get any deeper into that, but the interesting application to this is that when you take a little tube through which you're going to pass some light, whether it's plastic or glass or whatever, and you start the light down this tube, it stays inside the tube without losing any energy because it keeps reflecting internally in the pipe. Have you got that? Fiber optics. That's how they work. You can take that fiber optic and bend it around a pretty sharp corner, and none of the light gets out. It just keeps staying without any loss. It stays inside. Well, there's a tiny bit of loss, so every few miles you got to put an amplifier to take the signals and make them strong again. But that's the kind of the idea. Now, the application of that is that this is another drawing. There's the rods and the cones down here. That's a cone, that's a cone. Most of them are rods. They're just a drawing, but I'll show you how that occurs in the eyes. There is a cell that wasn't in the other drawings called a Mueller cell. And it spans the entire 150 microns, forgive me for that, of the saran wrap. And what it does is it catches the light up here and keeps it all inside this light pipe until it gets clear down here to where the rods and the cones are, and it gives them the light and they make their signals. Sounds like somebody knew about total internal reflection, doesn't it? Now, those are nerve cells. The Mueller cells are nerve cells, and so you see a lot of interesting connections here. And frankly, we're mystified by a lot of these things. There's just so much communication going on, folks, inside the retina to make this all work. It is mind-boggling. And the amount of calculations, if you will, that happen in there have been estimated in the billions per second. But nevertheless, let me give you one more thing with this other picture. There's a problem with, forgive me for the big words, something called spherical aberration. Did something fall down? Is aberration? It doesn't have two B's, does it? It has two R's. Or maybe one R and two B's. I think that's what it is. Okay. Spherical aberration. What that means is this lens is a part of a circle. You understand what I'm trying to say? And when the light comes in, it turns out I should get a different color, but it won't matter. The blue light bends more than the red light. Let's say this is blue, this is green, and the red light bends the least. So you have a fuzzy image. Got it? And if you buy, are you all with me on this? Is anybody, did I lose you on this idea? You know from Newton in his prism that light bends, the colors bend. And I'm telling you, and don't ask me why, I'll tell you, but you don't want to know. The blue bends more than the green, which bends less than the red, which bends more than the red. So you get this spread. I need to erase this and show you one other thing. Joe, if you'd stabilize this for me, it would really help. Okay, there we go. When you buy a nice expensive camera, you get this, they have to correct for spherical aberration in your camera, or the pictures would all look poor. And the way they do that is they make this out of a certain kind of glass, and so the light comes in here and it bends both places, but it bends this much totally. And then they put next to it, I'm going to put it further away, they put a concave lens in here tucked up against this one, which causes the line to straighten back out some. In other words, it bends back up instead of down. Got that?
The image isn't upside down.
No, not yet. And it still focuses down here, okay, at what we call the focal plane, which in this case is the retina. But because this is a different kind of glass, they can make those colors come back together so they focus. In an expensive camera, what you call the lens might have 5 or 6 pieces of glass in order to accomplish this correction. Got it? Now, interestingly enough, the creator, again, it'd be fun in class, right? Didn't try to make the cornea and the lens behind it have a shape that could correct for that. Instead, he made these Mueller cells, and the reason they have a wide top, you see it, is to collect all the colors. Remember, one is here and one is here, right? So they're spread out here, right? So the wide top catches them all and takes them in a light pipe down to the rods and cones. You got that? Mueller cells. Does this thing look like it was designed, by the way? Just incredible. Now here's an actual effort to illustrate this. These are the rods and the cones down here, and these are some American cells, and there's There's the ganglions and so forth. Anyway, this light spread out gets pulled down into this pipe, and then notice it spreads back out and whoops, wrong direction, and connects up with several rods and cones, so several people can get the same information. And this information is compared, and there's these on and off switches that are all going like crazy, and it's just the most amazing thing to me. Here's another couple of drawings of the Mueller cells, the whole width of the retina. Another drawing of it. This is light coming in from the bottom. There's the rods and the cones. There's this pigmented epithelium and so forth, and different cells doing their thing. Another one. Another one trying to illustrate all the connections, the nerve connections between all these different things that make it work. Now here's a diagram of some rods showing you the little discs. And notice this bipolar cell. They give it that name because this cell is able to in getting signals from other rods and cones. See, the rods and cones are all talking to each other with these signals. And here they come from these what we call horizontal cells. But the bipolar cells are making decisions as to whether or not to let that signal, that light signal, to get out of the cone and be transmitted and so forth. So it's a very complex system which nobody completely understands at this point. Lots of intensive work has been done to try to get some grasp of it. But at least we know the structure. We know that all these signals are being looked at and judged upon, whether or not they're going to be passed on and so forth. Now, a fascinating thing to me is that when piece of light, and physicists have a name for that, it's not important, but you know the name, it's a photon. How many of you ever heard the word? A lot of you. When a photon hits that disk, it actually uses up the disk, unless it's really a dim one, and then it might not quite use it all up. So every day in the retina of your eye, 10 billion new disks are made inside the rods and the cones to replace the used up ones every day. And if the light is really bright, it can even clobber, if you will, that's a real scientific term, it can even clobber the whole rod, and the rod will die and get replaced. And this is happening so constantly, and in fact the debris from the disc that gets used up, the disc breaks up, and the debris from that, remember now the light's coming in like this, and the debris from that, or from even a cell, although a macrophage will come in here and clean that up to some degree, but this stuff all has to be picked up. And that's why these cells are at the bottom. So when this happens, this lining of cells at the bottom, the pigmented epithelium, picks up this debris and cleans it out. And if these cells were far away, that debris would take time to get cleared out, and it would fog the image. Furthermore, have you all done this when you were kids? You get a magnifying glass and you go outside in the sun at recess when the teacher's not looking and you start a fire. Have you ever done that? This is light energy, folks, that's being focused, right? And it's a lot of heat on the back of the eye. And if the circulation is not good, the eye suffers. And I'm not a clinician, so don't ask me any questions about eye diseases. I know a little bit about some of them, but there is a huge issue in terms of the way you live and how healthy your eyes stay. Many of these eye diseases are the result of poor living. And this is again in another world, but I'm just going to mention it because it certainly connects with the eyes. Our circulation is compromised all over our bodies when we get too much fat, and the red blood cells that normally pass through, I actually have some slides of this. I guess I should put them in this section. The red blood cells that pass through the capillaries clump together. There's a name for it. There's a French scientist that discovered this, so it's called the rouleau effect. And the clump cannot get through the capillary. The average person in America who walks into Weimar, I have a physician friend that checked this for a few years when he was there. He uses an ophthalmoscope. You can look at the retina, and he estimates that about 80% of the average American's capillaries are clogged because of this red blood cell sludging or clumping. And when they put them on the Weimar program, active, walking 2, 3, 4 miles a day, put them on a plant-based diet without without refined products, within about 3 or 4 days, when he checks them, that clumping is down to 10, 20%, just because they changed their diets and became more active. And I was discussing this at length with an optometrist who himself is interested in this world very much. He just couldn't quit talking about the benefit to the eye. Now, usually we think our heart or something else, right? And it's been over a year ago now, a couple of years, when I was working for Dr. Neil Nedley. I used that name before. I'd love to take just 10 minutes to just tell you about him if you don't know him, but he's one of the most astute clinicians I've ever met in my life. I just am so impressed with this fellow and his dedication on top of all of that. But here he is. He's just an internal medicine physician. And I was down at a university where he went to medical school, and they had invited him to come speak to the ophthalmologists. There was about 60 to 70 ophthalmologists in a room, maybe half again is this big, but the big lecture hall. And he stood up there for an hour and a half, quoting to them from the recent literature. There's lots of it older, but he was quoting to them from literature, from studies that were done within the last 3 or 4 or 2 or 3 or 1 year, for an hour and a half, studies that show the benefit of lifestyle to the diseases of the eye. I just was amazed that they would sit and listen to somebody that didn't know anything about ophthalmology talk to them, see. Very, very interesting. I thought you'd want to know that. Even at your extended age, would it be a wise thing maybe to make some changes and help your eyes? Well, here is the blind spot, but if you went straight into the eye, straight to the back of the retina, there is a place where instead of being as thick as saran wrap, it gets to about a fourth or less as thick. And this is called the fovea. It has a couple of other names that are a little not terribly important. This is the part where we can see in detail. And it's very tiny. It's only 1 degree wide. Can you imagine drawing 2 lines with a protractor that was only 1 degree wide? Y'all with me? And if you go out there just that far, that's maybe a couple of millimeters wide. And the fovea, in order to get that kind of resolution, doesn't have this huge layer of all of these different things. It has enough to operate, but it's quite thin. And so the artist has pictured it. Can you see how the retina has become very thin right there? There it is, straight at the back of the eye. There's the optic nerve head where all the nerves are going. These are not nerves. These are arteries and veins that you see there. But that's where and actually in the retina, there's a little yellowish patch there. You can see very quickly where that nerve head is. That's the blind spot. But there's a diagram of the, in fact, when you get into about this portion of the retina, it's all cones. And as you move away from the middle of the fovea, you slowly start to see a few rods and more rods. I think you all know the cones detect color. That's easy. Cones see. The rods just detect black and white. In fact, there are 3 kinds of cones. red, green and blue, and we've only been able to prove this just recently, that that's the case. And that all has to be integrated with all this signaling in order to make the image. You know what I mean by all this signaling? All these nerves talking to each other and saying yes and no and so forth. And if you have your computer screen and you're typing in 12 point, 12? And you take a small eye and look at the dot. That's how big the fovea is where we see the detail. And in that dot on top of that 12-point eye are, got to think about this number now. It's not all that critical, but it's fascinating. 50,000 cones. 50,000 cones in that dot. Is that amazing or not? Just astonishing. And actually, you know what? This thing, okay, oh yeah, this graph is showing the acuity, the ability for your eye to discern between two fine lines close together, to see them as two instead of one, resolution, if you will. This shows the acuity of the fovea, and you can see it's hardly a degree wide where you get the maximum amount of that. Y'all with me on this? And it's, by the time you get out here to the blind spot, the acuity is measured here as 100%, and compared to that is kind of small. So there's another one. There's the fovea. That's the thing we were just talking about. And there's the nerve head as all the nerve fibers leave. There's an actual photomicrograph of the rods and the cones and the rods. And here's a drawing showing you all the discs. You see them? Photon comes in. The disc explodes. Well, it's not quite like that, but it's kind of like that. And so there's what? A whole stack of them, so you can keep using them, and the rod gets busy, or the cone gets busy making new ones to kind of keep up with the loss. And if you have snow blindness, it takes several days. You aware of that? We say snow blindness. This person isn't blind, but boy, their vision is really compromised because there has been so much light that some cells have died, and the discs are mostly used up, and it takes several days to build them all back into place again. Very interesting. Yeah, underwater. Real bright up near the surface. This is also an actual photograph using an electron microscope. And you can see the cone, and you can see the rods, cone, and so forth. This is the one I thought I was seeing. This shows you the density of rods, lots and lots and lots of them. Except when you get right to the fovea, no more rods at all, only cones. Does that blue line make sense, what I'm talking about? How many per square millimeter? I don't know if this is actual number. I think it may, or if it's in hundreds or something, but anyway, you can see that this is none, this is more, this is a lot. So there's a lot of rods, but less and less rods as you come toward the middle of the fovea, and that's where there's just lots and lots of cones, and no cones after you get out to about the blind spot. Can you see that? Blind spot's at about 14 degrees. In fact, that is the blind spot right there. And so by the time you get out here, looking, you know, about that far out, everything else is being done by the rods. Now here, the plot thickens and gets just fascinating to me. I just can hardly. This is the brain. Here's the two eyes, and this is the optic nerve as it leaves the two eyes. And there are, like I say, about 1 million nerve fibers in the optic nerve coming from each eye. And they come back in here not very far, just underneath the frontal lobe, toward the back of the frontal lobe. They cross, and half of the fibers from the right eye follow the fibers from the left eye. And half of the fibers from the left eye go along and join the fibers with the right eye and separate. Got that? There's a name for it. They call it the optic chiasm. And then the nerves, a third of these, and 2/3 of these, and at the same way, 2/3 of these, and a third of those, come to another synapse. You all know what the synapse is, right? It's an opportunity for a place where you can control the signals, because if you have another synapse nearby with some inhibitory synapsing going on, you all with me on this? Another place where signals can do interesting things that we don't fully understand, and then they all travel back here to what's called the visual cortex. The main part of it is right in the occipital lobe. I'll show you another picture, but there's a whole region of the brain back here where these nerve cells, these nerve endings go, but it's worse than that. At this point, one of the reasons there's a synapse there is because a lot of connections will go to various places in the brain that have nothing to do with forming the image, but all over in the brain there are places that need to know what's going on out there, what you're looking at. You got it? Now here's the diagram. This is the front of the brain. Back here at the occipital lobe is where the primary visual cortex is. That's where the vision is mostly put together. But there are these secondary visual areas. And the way we know that, folks, is we put people in a dark room, we put all these electrodes, or we use what we call a functional MRI, and we flash onto the screen a color or some kind of a image, a simple image, and we watch to see where biochemical activity occurs. What does that mean? Cells are doing stuff, and you can see it happening. So when people look at things, and we have mapped the brain, I told you this, to within about 1 millimeter, every place in the brain we know where stuff happens because of watching biochemical activity when we limit somebody's doing stuff to just something. Y'all with me on the idea? And so that's why we can say all of this area is part of making the picture, the image, and yet in other places the nerves go and we're not just sure what they do there. The visual cortex is organized into a primary and secondary region in each occipital lobe. Direct visual signals come into the primary cortex, which is located in the occipital region. The fovea, the region of the retina with the highest visual acuity, sends signals directly into that and is heavily overrepresented there. What does that mean? Of all those nerve cells that are coming out of the retina, the fovea has most of the representation there from the nerves from the fovea. You understand, folks, nerves are coming from all parts of the retina, right? All parts. Compared with peripheral retinal regions, the secondary visual cortex receives signals secondarily. They are transmitted to these areas for analysis with respect to motion, shape, position, etc. via intracortical pathways. Cortical means cortex, the brain itself. And what scientists have learned is that the eye is just, it's doing amazing things. One thing it does, it just draws outlines of things. And if it starts to move, then other signals add to it that this shape is moving. It's just astonishing what's going on in there. And here's the interesting thing. We do not have a clue how the image is actually presented and formed in there. All we know is there's a bunch of clicks going on. Is that what we learned the other night? A bunch of electronic clicks. It's amazing. Absolutely amazing. Different regions of the secondary cortex are responsible for different types of classification and analysis, and depending on the conclusion reached, actions can be initiated by motor control areas of the cerebrum. Now, when you get old and you have a pointer like this and you try to hold it on the screen, it's very hard to make it hold real still. You understand that? You want to try it? So it's not exactly a tremor, it's just the way the muscle is under constant control, folks. Constant control, laterally and vertically, if you wish. And those signals have a little bit of variation in them. Now, interestingly enough, for a muscle fiber, for a muscle nerve, there's about 10 fibers. For the muscles that control the eye, there's 10 times that many. Over 10 times that many. The muscle fibers. So that when you move your eye, it can move quickly to a spot, and the spot is not trembling like the red dot is. Y'all with me on that? In other words, the control is refined, it's made finer because of additional circuitry keeping it honed in on one spot. Is that cool or not? And in fact, the muscle fibers that run the movement of the head are also like that. Because if your head was twivering around, then the image would also shake, wouldn't it? Isn't that amazing? Just amazing. All right. Believe it or not, this is the closing story. When I was a young science teacher, I'm stopping just for a second. I don't have notes on all this, and I'm just trying to remember what I may have left out, but I think we did pretty well there. Okay, when I was a young science teacher, I learned about some films that were made by the Moody Bible Institute by a fellow named Irwin Moon. And he developed these, and in those days they were on 16mm film. And I would rent them and show them to my science classes because he did a lot of very interesting things in science, and then he drew spiritual lessons from them. So they were called Sermons from Science. How many have ever remember seeing one of them? Whew, not very many. Oh, that's too bad. You can buy them today. You should get them and show them to your kids and show them to you big kids sometime for some kind of meeting. They're very interesting things. Well, in 1974, there was a World's Fair. I got to back up. Of all the things that I saw Dr. Moon do, and he did a lot of interesting things. One that I remember was he got a rattlesnake all tied up here in a thing and blindfolded it and got some detectors hooked up to its infrared sensors. And those detectors were amplified so you could hear noise. You bring something warm in front of the rattlesnake's face and this noise in the speaker, you know, and just your hand. You know, y'all with me? Well, the thing that of all of the things that I was fascinated with, because most of them had to do with the physical world, and that's interesting to me, was that at one point, to try to illustrate what a marvelous brain we have, he had his lab build a pair of something that looked like a little bit like binoculars, maybe about this long, just a little longer, and there was a kind of a cap strap thing that held them in place, and he saw the world upside down, inverted and left for right. And for 6 weeks, he never opened his eyes without those being on. He would put a mask on and sleep with it and so forth. And at first, even sitting at a table, somebody would roll a ball to him and he just couldn't even begin to get it. And he just reached in the wrong place and it was just crazy. But after a time, here he is riding a motorcycle in in a what? A maze or whatever you want to call it, an obstacle course. And he's a pilot, and he's flying an airplane, and he's all the time, he never opens his eyes except doing this. Y'all with me on this? Well, I'm really amazed about that. But that's the end of that story, except in 1974, there was a World's Fair at Spokane, and I was asked by the North American division to take care of the big booth they had put up there. It was full of electronic stuff, and every day I would drive into Spokane. I had a pass. I'd go in there and check all the stuff, and if something wasn't working, I'd try to fix it. Well, because Moody Bible Institute had a booth, not a booth, but a pavilion, if I wasn't too busy, I'd slip over there and watch one of those programs. And I was sitting there one day. It was a room about like this. And I'm sitting over here, and there's a guy walking around up here taking pictures. And there was something familiar about this guy. And it dawned on me all of a sudden, that is Irwin Moon 20 years later, from when he did all these films. And so I want to talk to him. So I got up and walked over there when he was kind of not busy for a second, and I introduced myself, and I said I'm a science teacher at a high school nearby, and I use your films all the time. Could we just visit for a few minutes? He said, sure. We went outside and sat down on a bench, and I told him that I was especially interested in, it's in a film or a DVD called Windows of the Soul. And he's talking about our sensory input and what it means in terms of spiritual issues. He always has these marvelously interesting demonstrations, and then he draws these lessons. So I sat down with him, and by the way, he passed away in '86, and one of the things he used to do, he would get on top of a Van de Graaff generator and get 1,000,000 volts going, and this is just, these are the names of all but 2 or 3 of the films that, and they're now available on DVD, that he used to do. Anyway, so this isn't a terribly important part of the story, but he misunderstood me when I mentioned the Windows of the Soul. He thought I was going to do this with some of my physics students, and boy, he really he was terribly agitated. And I said, no, I just want to ask you some questions about, what you did. So this is what he told me. He said that after a few days, it became completely normal for him, and he could not tell that he was looking at things upside down. So it wasn't a problem to drive the motorcycle through the obstacle course. It wasn't a problem to fly the airplane to fly the airplane. Well, that was very interesting to me and to him and to anybody, because the brain was able to adapt to this crazy situation, see? And he said, if I walked into a room and there was a picture hanging right side up and another one upside down, the exact same picture, he said, I wasn't sure which was which. But it was working for him. It was just natural, and he could just function normally. Now, there's an interesting issue here. Back in those days, they might have found somebody that could have done better, but he was actually seeing kind of tunnel vision with these, you'd kind of look around a lot, but it still was working. So it came time after 6 weeks to take these things off, and he said a couple of physicians that were working with him on this came in there, and they took them off, and he said within 30 seconds, I was a blithering idiot. It's the words he used. And what was going on, they realized, and of course as soon as they saw him start to fall apart, they quickly covered his eyes and took him into a dark room and had a little conference and figured out that what was going on apparently was he was exposing mostly the rods for these 6 weeks. And when he took this off, the brain gave him the regular image from long ago, which was upside down and inverted to the image he had come to be used to, like this. Y'all with me? And it took them several weeks, put him in the dark room, and they brought the light up just a little bit so he could just barely see with his rods and not the cones. I didn't say this. I think most of you know that the rods are not as sensitive as the cones. When you go out at night, the number of cones becomes very small as you get very far out, and you're mainly looking at the night sky or wherever you're looking using the rods. Did I say that backwards? The number of cones gets small as you go out here. And so they took advantage of that to slowly try to retrain the rods while the cones were doing the old thing they used to do. Which just, I tell the story just to illustrate another amazing thing about what's going on in our brains with all these signals and its capacity to do amazing things with all of the gifts that we've been given in our marvelous bodies. Well, that brings us to our close, except I was hoping that all week long you were doing some thinking. And you know, why would I want to come and do this? I want to encourage people to have a deeper trust, not only that our bodies were designed and created, but that it was by a loving designer. And for those of you that are interested, many of you are worshipers, but for those of you that are interested, I hope that if this isn't where you've been in recent parts of your life, that it would make you say to yourself, this is very interesting. Apparently somebody designed me, and that, it seems to me, friends, would elicit two things from our thinking. One is it brings responsibility. Is that right? And the other thing is, it raises the question as to whether I would want to worship this God, if that's what it was. And that's what I believe, of course, is that I don't know how any person with human capacity, folks, could ever design and build this kind of stuff. Would you agree with that? It's just too far out there. And I just hope that most of you who are believers in God, I just pray that you will be deeply moved, perhaps in ways you haven't been before, about the marvel, and that I want to be more serious about my responsibilities to the designer. I'm in awe. Are you in awe? I'm in awe. And I recognize that from just this, we might not know very much about that designer. If you consider the Bible as a source of information about the designer, what you also learn is that he loves us. He wants to protect us. He wants to save us and have us live with him forever. He wants to bring an end to the sin in this world. And I just pray that our time together this week will have accomplished those kinds of things in your thinking. Make you want to praise God more. Make you long to serve him more wisely, more carefully, more according to his plan. And also, friends, you all know this. I'm sure you know this. For those people who don't know him, I want to help them come to a place where they're interested in him too. Amen? Let's pray. Father, I thank you personally for the evidence of your amazing creative hand And here we are, we know, Lord, we are just scratching the surface. Astonishing, marvelous, incredible almost, what you've given us. We want to, I think I speak for us, Lord, all. We want to be responsible. We want to use this wisely. We want to take care of what you've given us. And be a good steward, not only of our physical bodies, but perhaps to be able to help others to see beyond what most people are thinking about in terms of their own lives. Bless us in that effort, I pray, in Jesus' name. Amen.